While we get ready for the science sessions to start up again, I just want to remind people of a couple things. First of all, the email with the URL for tomorrow's conference went out at noon Pacific Daylight Time today. So you all should have it. If you don't have it, if you didn't receive it, please check your spam folder. And if you still don't have it, please contact Jen, who's been running all of this and doing a great job of it. Her email, I'll pause in case people need to write this down, is jenhajjevents@gmail.com. If anyone didn't get that, please just type something into the chat and we can give it again. But you should have the URL for tomorrow's conference. And then also, everyone, please remember that the abstracts and the bios and the contact information for all the science session presenters is at the bottom of the agenda page on the conference website. There's a lot of good information there on these speakers and their presentations, so please check that out. And with that, let's go ahead and get started with the afternoon session, or the late afternoon session. Our next speaker is WFO board member Bryce Robinson. Bryce is an ornithologist and illustrator who has studied birds across North and Central America. He's the author of a number of scientific publications, some of which feature his illustrations. And Bryce lives in Ithaca, New York, where he's working toward a PhD at Cornell and the Cornell Lab of Ornithology. And today, Bryce will be speaking about red nuchal markings in red-shafted northern flickers. So Bryce, welcome.
Thanks, Steve. Hopefully everyone can hear me okay. I'll get this screen shared. That looks good. As Steve mentioned, I'm just finishing my time on the WFO board, but I'm also a PhD student at Cornell University. And I'm going to share some work I'm doing, kind of a little side project I've been focusing on lately, looking into the prevalence of nuchal markings in red-shafted northern flicker. So to start, I thought, shoot, not switching for me. Thought I'd start by giving an overview of the distribution of red-shafted or northern flicker in North America. Most of you probably are very familiar with this distribution, but for those of you that don't know, in North America, there are two subspecies of northern flicker. The dominant auratus in the east, which is also called yellow shafted because of the coloration of the flight feathers. In the west is the subspecies coffer, which is also called red shafted flicker because of the coloration of its flight feathers as well. Where these populations meet, basically along the continental divide, they come into contact and they breed freely. They don't treat each other as different. And that creates a broad hybrid zone where there are individuals that have characteristics of both parental types. And I thought it would be pretty important to go over the identification of both of the subspecies apart from just their flight feather coloration, but also give an explanation of how to identify a hybrid, or what we call intergrades for subspecies pairs, as the basis for the rest of the stuff I'm going to be discussing. So the yellow shafted, or auratus, has a red nuchal crescent and of course yellow flight feathers, but it also has a gray crown with tan cheeks and a tan throat, and then males have a black malar stripe, as you can see in that illustration. The red shafted of course has these salmon colored flight feathers, but also differs from the yellow shafted in having a brownish or tawny crown, gray cheeks, a gray throat, and then males have a red malar, so quite different from auratus. Intergrades, they can be identified by basically a combination of traits from each parental type, and they vary quite a bit. But I've tried to summarize what one intergrade might look like in this illustration Many have red nuchal crescents. And then crown color generally is one of the parental types, but it can also be mixed between brown and gray. And that the same as with the cheek color and the throat color, can be mixed or one of the parental type colorations. The mallard is generally a mix of red and black, and that often stands out quite a bit. And then the flight feathers, they can be one of the other parental types, or often they're yellow-orange coloration, which is essentially a mixture of the two colors of the parental types. During my time as a younger birder in the West, I did notice birds, red-shafted flickers during the breeding season that had some red in the nape, noticeable red in the nape. And at the time, I didn't understand exactly what that would be, but from talking to some folks, I was told that that's just something that occurs rarely, but it does occur in red-shafted populations. Later, as an eBird reviewer for a few counties in southwestern Idaho, I started noticing that folks were putting these individuals, these otherwise red-shafted flickers with red in the nape, they were inputting them into eBird as hybrids or integrades auratus and Kaffir. And the way I treated that as an eBird reviewer was to ask them to not put that designation on these individuals, because we know that they breed within the red-shafted distribution, so it doesn't really make sense that they're intergrades. And that was based on my personal observations. Understandably, I did get some pushback on that, and so I decided that I should dig into it and try and get some data and see exactly how prevalent this trait might be. So I decided to source photos in the Macaulay Library, and basically what I did was I selected photos for 7 states that are within the described distribution of red-shafted flicker. I did not look into states that contained the described hybrid zone. And I only looked at individuals during the breeding season, so from 1st of May to 31st of July, which is a rough breeding window. And I should note that everything I'm presenting is up until 2022. I haven't looked at 2021 photos yet. But from this photo pool, I recorded individuals that had any amount of red in the nape, that were otherwise red-shafted flickers. My computer's lagging. So I just want to walk you all through how I did that, so that you can yourselves go and do the thing and see if you agree with what I found. So I this one page and just typed in northern flicker and selected northern flicker. And that's it out.
Hey, Bryce, sorry to interrupt. This is Steve.
Just over 100,000 photos. So wait, and
you're breaking up, and I don't know if there's anything you can do about it on your end, but you're breaking up.
The photos from photos.
Breaking up, and I don't know if there's anything you can.
Yeah. Can you hear me now? Or is it...
Bryce, I'm going to turn off your video, and hopefully that will help.
Let me stop sharing and see if I can go.
Sorry about that. We're working on it, and we'll see if we can get this started up again where we left off. Thank you for your patience as we work it out. Hi, Bryce. Are you still there? Bryce, are you still there?
I'm here. I can hear you now.
Hello, folks. I'm very sorry that we're experiencing some technical difficulties. I'm going to make the recommendation that we move on to Lucas's presentation and then come back to Bryce and see if we can see the rest of his presentation after we've presented Lucas. So I hope that Lucas, you can be ready to be on deck. And Steve, if you would like to make the introduction, we can move on ahead.
Sure. Sounds good. And every online meeting I've had for the last year and a half has had one glitch. So this is the one, which is good. Get it out of the way now.
I hope so.
Yes, but yeah, Luke, thanks a lot for stepping in. So want to introduce Lucas DECICCO. Luke is a PhD student at the University of Kansas. He has a long-standing interest in birds and their interest-specific geographic variation in plumage, vocalizations, and more recently, patterns in genetic variation. He is motivated by understanding and documenting patterns of biodiversity through museum-based work and evolutionary research. And today, Luke will speak with us about a contact zone between two fox sparrow subspecies groups in South Central Alaska. Thanks a lot, Luke.
Excellent. Thank you, Steve. Let me share my screen. All right, does that look good? Can everybody see it? Thumbs up? All right.
Yes.
Okay, I'll keep going until I hear any complaints. Thanks for the intro, Steve. I hope I don't have internet issues as well. But yeah, let's get into this. My name's Luke DECICCO. I'm now a PhD student at KU. And today I'll be talking about a breeding contact area for fox sparrows in South Central Alaska between two subspecies groups. Breeding contact areas are inherently interesting to naturalists and evolutionary biologists, as at these sites or regions, we can observe how different taxa, be it differentiated populations or species, interact naturally on their breeding grounds. And that varies widely, basically depending on a lot of things, but mostly breeding isolation or not. So we're going to start with a bit of review on fox sparrow taxonomy. Here and throughout the rest of the presentation, I'm strictly talking about breeding distribution. So all or most populations of these fox sparrows are migratory. They all have wintering distributions, which are different. But this map and the rest that I'm talking about is only talking about breeding distributions. So unlike many groups of birds that we're familiar with, fox sparrow subspecies, and there are approximately 19 of them, give or take, they're broken up into 4 subspecies groups. So essentially underneath the species level, there are 2 levels of organization. And ornithologists have done this because, well, one, it's a very complex system, but primarily because the described subspecies within a group appear more similar to themselves than to subspecies in a different group. And some people have recognized these as species. We're going to stick with the common thinking that groups of subspecies within the fox sparrow. Okay, we're going to start off with the Iliaca group, which are commonly referred to as the red fox sparrows. They are distributed across boreal Canada and into interior Alaska out to the western coast. The Unalacensis group, or sooty fox sparrow, occurs along the Pacific coast from the Alaska Peninsula throughout South Central and Southeastern Alaska and into Washington, or through BC and into Northern Washington. The Schistaceae group, or slate-colored fox sparrow, occurs in inner Rocky Mountain west of the US, interior British Columbia, and then a few isolated populations into the southern US states. Phenotypically, the Unalacensis group is really brown and drab. Iliaca group is well marked and reddish. The Schistaceae group is relatively well marked and overall grayish. And the last group we're going to talk about is the Megaryncha group, or thick-billed fox sparrow. Plumage-wise, similar to Schistaceae, but has a larger bill. It's distributed in interior Oregon and California, essentially. So focusing on kind of the more topic that we were talking about, what are our expectations for breeding connectivity within these subspecies groups? So between subspecies and then between these subspecies groups. This is roughly based on species concepts and subspecies concepts. Within group, so between or among closely related subspecies, we would essentially expect almost continuous variation and really no isolation, breeding isolation, where these taxa come into contact. We see that in Alaska in the coastal Unalacensis group, whereas you move along the coast from the Alaska Peninsula into southeastern Alaska, you essentially see relatively continuous variation. At least you don't see any hard breaks between the subspecies. However, between these subspecies groups, we don't really have a set of expectations for what we should see. Presumably, we'll see less free interbreeding than among subspecies, but we really don't have a clear set of expectations. Let's focus on Alaska now, and the rest of the talk will be geographically focused here. In Alaska, we have 2 of the subspecies groups are represented. Iliaca group represented by subspecies Zaboria, and 6 subspecies within the Unalaskaensis group distributed along the Pacific coast. There are 3 or 4 known areas of contact between these 2 groups. It's not a broad geographic region of contact due to the coastal mountain ranges in Alaska that broadly really separate the coastal Pacific rainforest habitats from interior boreal forest. And we don't really know much about many of these contact areas. The first contact area is this relatively broad and complex region on the Alaska Peninsula. Williamson and Peyton described it roughly in the mid-1960s and essentially described it as a contact area between 2 groups, and it involved at least 3 subspecies. There's obviously a lot of work to be done there. It'd be quite interesting. There's a very narrow contact area in the Anchorage area, also recognized by Williamson and Peyton in the mid-60s, but really not much has been done on it at all since then. And this is the area that I'll be talking about today. And there's 1, but probably 2 contact areas to the east of that in Prince William Sound and the Copper River drainage. And as far as we know and areas that have been published upon, these are possibly the only contact areas between these 2 subspecies groups. It's very likely that there are overlooked areas in southeast Alaska and in BC. But this is what we're going on based on our current knowledge. So the Anchorage area is circled here. This is the contact area that I looked at and that all the data I'll be presenting today come from. To give you a bit of perspective, you can see the city of Anchorage is here. You have the foothills of the Chugach Mountains here and some lowland boreal forest here. Just south of Anchorage on the Kenai Peninsula, basically just south of Turnagain Arm here, you have only coastal Unalaskaensis sooty fox sparrows. And north into the Kenai Mountains here, over to the west or east, you have only Zaboria or red fox sparrows. How broad this zone of intergradation is uncertain, but it's quite narrow. And I'll be talking about what I observed right here in and around Anchorage. So the information I'm presenting comes from opportunistic work done over 4 breeding seasons. Fox sparrows they occur in very small numbers, very rarely, but they basically don't occur outside of the breeding seasons in Anchorage. But my focus was during the breeding seasons and on territorial birds. So my information is highly biased towards males because they were easily detectable and I could collect the information I wanted off of them. So the documentation that I took on territorial birds that I found were photos in the field, audio recordings, and a selection of specimens to document the variation I was observing. Over the course of those 4 years, I documented well over 60 territorial birds in the Anchorage area. And basically the take home that I observed was that the parental phenotypes were represented at approximately a 1-to-1 ratio. So roughly over a certain given area, you could find approximately the same number of interior Zaboria birds as coastal sinuosa birds. And that birds that were phenotypically intermediate were quite rare. Further, there was really thorough spatial mixing of the taxa. It wasn't like you had sooty birds in one area and then, in the foothills of the Chugach Mountains, maybe you had interior birds. This map is a subset of the territorial birds that I detected and documented, and it overrepresents intergrades, as I was really focused on trying to document that phenotype. But you can see, so this area is really the core of my research area, and you can see representation of the really thorough spatial mixing here. So black dots are coastal birds, white interior, gray intermediate. The plumage aspect of these birds varies a lot. These specimens represented here were all taken at the intergrade zone in Anchorage. On top, we have a representative of the subspecies Zaboria, the Iliaca red interior group. Phenotypically, it appears to be pure Zaboria compared to birds in interior Alaska, and it looks identical. Down here, we have the coastal subspecies Sinuosa of the Unalakensis sooty group. And you can see easily the very relatively drastic plumage differences between these two, particularly in the facial markings, with Zaboria having a very well-marked face, lots of gray and red, Sinuosa having a very plain face. Similarly in the breast chevrons, and particularly in the wing coverts, with it being very plain and brown in Sinuosa, and well marked with different colorations and patterns in Zaboria. This middle bird represents a phenotypic intergrade, and basically shows intermediate characteristics in plumage in all these aspects. Similarly, their dorsal plumage varies quite a bit. The coastal sooty birds have a very clean brown back with a dull, slightly redder tail and rump, compared to the interior Zaboria birds, which have a very well-marked streaked back and a pretty bright rump and tail. And the intermediates or intergrades are cleanly intermediate between these two. Their songs also differ a lot. I think there's like a tremendous amount of work to be done on the vocalizations, and I'm just giving you guys some examples here. I have 2 recordings here. Hopefully they'll play and work. So the first set are of a Zaboria bird, interior red bird in the contact area. Their songs are very languid, clean, clear notes, and they have high consistency both within individuals and between individuals. So this recording is of one individual singing 2 songs consecutively. And I only heard one song type, and these birds would repeat it time and time again. So basically the same song type. In comparison, Sinuosa, or the coastal sooty birds, have a very choppy, kind of abrasive song, and they sing multiple song types. So you see both variation within an individual and more variation between or among individuals. This recording is of one individual that sings 2 consecutive song types. And you can hear that trill at the end. That is a portion of the song that is entirely lacking in any Zaborias that I've documented. What I find as one of the most interesting aspects of this is their habitat preferences, and they have clear habitat preferences. Zaboria, the interior boreal forest bird, prefers boreal forest. The Sinuosa coastal bird prefers alder thickets. And it's basically in this entire Anchorage area, whether it's lowland boreal forest, mixed boreal forest, or the forested areas on the slopes of the Chugach Mountains or their foothills, you have this mosaic of these 2 habitats. Alder thickets often grow in recently disturbed areas. They're a very early successional species. But it's a common forest type in the anchorage area is this mosaic of boreal forest and alder thickets. And I really believe that that's essentially what's really causing the heavy spatial mixing between these two taxa. This photo represents a very typical forested area that you might find in that area region. The background is what I would consider mature mixed boreal forest. I would not be surprised to have a Zaboria bird holding a territory here. And this foreground shrubbery are alders. And it happens time and time again where you have adjacent birds holding adjacent territories that are representatives of phenotypically pure Zaboria or phenotypically pure sinuosa. And this photo represents basically that habitat mosaic. So take home is that I observed really thorough spatial mixing of these two phenotypes. So how does this fit into kind of our expectations of what we might see for breeding contact between two taxa? This diagram is highly simplified, but hopefully helps explain what I would expect. So under one extreme of absolutely no reproductive isolation, which fits actually nicely with what Bryce was presenting and what hopefully we'll hear the rest of soon in the flickers, is that as you move from one population across a geographic scale, one population, say population A, of all pure individuals of one taxon, then you move across a geographic scale into all pure individuals of another taxon. And across that gradient, where you see intermediates, you see a smooth climb of intermediate phenotypes from one pure extreme to the other pure extreme. And importantly, anywhere geographically that you sampled along that contact gradient area, you should see a relatively homogeneous group of birds, but they should all show an intermediate phenotype. Now, at the other extreme, complete reproductive isolation, you could consider this as just two starkly different, well, not different, because yeah, two well-formed species. So take, say, golden crown and white crown sparrows, although I know that there are hybrids rarely. But anyway, if you have those two taxa or species contacting in an area, you would assume complete isolation. You could either have abutting distributions or overlapping distributions. But if you had overlapping distributions, you would find both species at these contact areas and no intergrades. And then there's a whole continuum of intermediate stages. And I believe that's where this Fox Sparrow system kind of lays, where when you have a contact area, you have partial reproductive isolation. So they don't freely interbreed, but they do interbreed and produce phenotypically intermediate individuals. So at any site in this hypothetical, this intermediate one, in any geographic location in this contact area, you would expect to see 3 or possibly more, but 3 phenotypes at any one area. The 2 parentals and presumably few intermediate phenotypes. So that's kind of where this system stands. And my take home of my understanding based on my observations and data is that these two taxa do not freely interbreed upon breeding contact in anchorage area. And there is therefore some selection against interbreeding. And it could be prezygotic. So if there's positive assortative mating based on plumage or song, or postzygotic, where the intergrade offspring are selected against. There's a whole lot of cool stuff that could be done with these contact areas. We know very little. Well, we know basically what I've presented. We don't know where the geographic limits of this contact area are. And we know almost nothing about other contact areas nearby between the two same taxa. I'm really curious about the genetic patterns at this contact area, if they reflect the phenotypic patterns that we saw or not. And that's my next step. And it's actually moving forward to get genomic data from the sampling in Anchorage. A couple cautionary notes. This is just one contact area. So my findings from here should not be extrapolated to describe contact between these groups or other groups in all contact areas. What I've presented so far is based on the phenotype only. We don't know yet the genetic patterns, but hopefully soon. And something that Phil Unit brought to my attention and that we've discussed in other publications is that these intergrades appear very similar to other described taxa. So the intermediate fox sparrows, intergrade fox sparrows from South Central Alaska appear likely almost identical to the subspecies Altavagans from interior British Columbia. This clearly isn't a problem on the summering breeding grounds, but it is a problem and something that should be considered when trying to identify fox sparrows on the wintering grounds. And this is a very similar pattern that we see in orange-crowned warblers and dark-eyed juncos, where intergrades, hybrids, or intergrades from South Central Alaska look phenotypically almost identical to described taxa from interior British Columbia and inner Rocky Mountain west of the US. With that, I'd like to thank a number of people and thank you for your time. And I'd love to try to answer some questions. So, yeah.
Thanks a lot, Luke. Let's see. We don't have any questions posted yet. I'll just say, oh, okay, here you go. From John Dunn. What is your suggestion? Ready for a motion to the NACC? As I recall, Zink and others said that Iliaca was the most distinct genetically based on mitochondrial DNA.
I thought someone would ask that question. I'm not surprised it was you, John. I don't know. I will have good genetic data really soon for a lot of these, and I'm really curious what that says. I'm guessing that that's also not going to clearly answer that question either. I would say though that in Anchorage they act more like species than our typical semblance of subspecies, based on lack of free interbreeding. But yeah, I think looking at contact areas between other groups beyond what Zink did would also be good. It's a cool system.
Another question, do intergrades only pair with another intergrade?
That's a wonderful question. And I wish I knew the answer. An unfortunate part of my kind of opportunistic work up there was that I couldn't spend a lot of time. So I have really very little information on the female of a mated pair. The males are always out singing. They respond to playback, very easy to observe, difficult to find or observe the female. I don't have any observations of the pair from an intergrade. However, many or all of the birds that I observed a pair were of the same taxon. So reds were mating with reds, sooties with sooties. That's a very small sample size, but I think it would, yeah, that's a wonderful question.
Paul Lehman says there are a surprising number of intermediate birds in fall on Alaska offshore islands, such as at Gamble. Have you seen photos and do you have any idea what they are?
Yeah, I agree with you, Paul. We also saw a large number of intermediates on Middleton during fall migration. I would assume that they're coming from these contact zones, and possibly if that Alaska Peninsula contact zone acts differently than the Anchorage one, it's much broader. There's no real good geographic barrier there. It's entirely possible that that Alaska Peninsula contact zone could be producing a lot of intergrade individuals. And on Gamble, I don't know. I mean, you guys could be pulling birds from reverse migrants from BC, I assume, too. So
Another question, what are the songs of the intergrades like?
So I've got recordings of I think two intergrades, 2 or 3 intergrades, and one sounded like Zaboria, clean Zaboria, and another one was mostly Zaboria, but had a little bit of sign of Sinuosa. So these guys, I mean, they learn their songs, so presumably they're learning from either their parental father or a nearby neighbor. So it's not surprising to me that you don't see intermediate songs in these things, since the majority of the birds there are breeding appear to be pure of either parental and present that parental song. So yeah, I wish I had more than just a couple from intergrades.
All right, and last question, can you tell red from sooty call on call notes?
Ooh, I can't. Somebody else with a better ear probably can.
That was John again, John Dunn. He probably wanted that answer from you.
Yeah, and I don't have recordings that are good for that either. All my recordings from that contact area are songs or, really excited calls due to playback. So yeah.
All right. Well, Luke, thanks a lot for that presentation, and good luck sorting all this out. We look forward to seeing it get sorted out in the future. So thanks a lot. We really appreciate it.
I wouldn't hold your breath too strong.
All right. Well, thanks a lot. Hopefully, Bryce, we're gonna try again. You got well into to the presentation before things started to break up. So if you're able to try again, that would be great.
Okay, can you hear me?
I can hear you just fine.
Yeah, I can back up a little bit, see where we are and see if it works. Okay, let's see. How about here? Does that make sense for us to pick up here?
I think it might have been a slide or two before this. Right there you go, right there. Right there was where it kind of broke up. Thank you.
Okay, what I was explaining here was that years ago, I for my birding in the West, I had noticed that there were phenotypically red-shafted flickers that had some red in the nape. And, this was during the breeding season. And I asked some people about that. I think Brian Sullivan or someone asked him if that was, just something that occurs every once in a while in red-shafted flicker. And he told me that it does. So later, during my time as an eBird reviewer for a few counties in southwestern Idaho, I encountered instances where people were inputting these otherwise red-shafted flickers, but with red in the nape, as intergrades, Auratus and Kaffir. And I would ask them to lift that designation off of these individuals because we know that they breed at least at low frequency within the distribution of red-shafted flicker. And, that was based on my personal observations, and I understandably got some pushback from some eBird users on that. And so I decided to really dig into it a little bit more. And I chose to do that through the Macaulay Library, just looking at photos to see if I could come up with some better numbers or a better perspective on the true prevalence of this trait in red-shafted flicker. So I chose to look at photos in 7 states that are within the distribution of red-shafted flicker, and I did not look at states that contained any of the contact zone. And I only looked during the breeding season, which I just binned the photos from 1st of May to 31st of July. That's a rough bin. And I also should say that I only did this up until 2020. I haven't looked at 2021 photos yet, but I'm sure that there are individuals in that photo pool as well. And from this photo pool, I recorded individuals that had any amount of red in the nape. And I want to go through and show everybody what this looks like, or how I went about doing this, so you yourself could go into the library and explore this and find some of these birds yourself. So if you're not familiar, this is a homepage for the library. I just put in northern flicker and selected northern flicker, no subspecies designation, and that is like again, and that spit out over 100,000 photos. And from there, I applied the state filter under the location tab. First, I started with Washington and went through, and I only did this one state at a time. As you can see, that filter knocked down the number of photos to just over 4,000. So then I applied the
It looks like we've run into some technical problems again. So I'm very sorry about that. It was very interesting presentation. I'm going to shoot it over to Steve to wrap things up.
All right, thanks, Jen. And Bryce, I'm so sorry that we're having issues, but I know that you have an upcoming session at 5. So as much as I'd like to hear about the flicker issue, it's probably even better for you to see if you can figure out that issue between now and 5. So to everyone participating this afternoon, thanks a lot for tuning into the science sessions. We're going to be doing it again tomorrow at 12:30. So we look forward to more talks then. And maybe if we can get things worked out with Bryce, we can hear the end of it tomorrow or Sunday also. But
that's a great idea. Yeah.
Anyway, so yeah, I'll coordinate with him. But for now, I want him to try to get things figured out for this session 5, illustrating birds from the field to the studio, coming up at 5 o'clock. So hopefully folks can tune back in for that.
It is my understanding that he is working on the problem and it should be much improved.
Excellent. All right. Thanks.
All right. We'll see everybody at 5.
All right. Bye-bye.